Okay, part 2 of the blood vessels, starting with arterioles. So we can see that from your outline, we can control the arterioles. Intrinsically, we can control the flow of blood, and we can control the distribution of the blood into the organs. And we can also control it extrinsically with our nervous system and with hormones. So let's start with intrinsic. Well, actually, let's start with looking at this picture. So we can see that, again, this is a same kind of image as we saw earlier. This is our heartbeat, right? So we have our pulse pressure. So we have pulsatility, which is the term pulsatility in our aorta, because we can feel the systolic and diastolic pressure. We have pulsatility in the elastic arteries. We have pulsatility in the muscular arteries, and a little bit in the arterioles, but that's when we start to lose it. And then so we can see the arterioles, right? At the beginning of arterioles, we might have a little bit of pulsatility, but at the end, we start to lose that. And then we have no pulsatility at all in the capillaries, venules, and in the veins. So in all through this, right, what we mean by mean arterial pressure, this is what we call blood pressure, right? Your mean arterial pressure, this is the average pressure between systolic and diastolic, is that middle line, that gray line. So hopefully this gives you an idea of when we take our blood pressure, we're taking our mean arterial pressure. So let's take a look at how your blood flow is controlled. And this is mainly, again, through your arterioles. So we saw earlier that we can change the lumen size, the radius of the arterioles. So at rest, we're going to have tone. Remember, tone is just a small contraction of all your muscles, even at rest when nothing's moving. And then if we have contraction of the smooth muscles, it causes vasoconstriction. If we have relaxation, it causes vasodilation. So again, constriction is going to increase resistance, right? And then if you dilate, it decreases resistance. All right. So looking at what controls the dilation and constriction of the blood vessels. Number 1 is active hyperemia. So this is basically where you are using the tissues. You're exercising, or your tissues are very active and they are using oxygen and they are creating carbon dioxide. And so we can see that we have these gases here, carbon dioxide and oxygen. And under normal conditions, if you're not really cranking up your metabolism, you're going to have, in this letter B, steady state, right? So you're going to have oxygen is delivered as fast as it's consumed, and then the CO2, carbon dioxide, is removed as fast as it's produced. So you kind of have this even flow, oxygen and carbon dioxide, and not one of those gases are building up. So hyperemia, just to go back to this word, right? Hyperemia is an increased blood flow. So hyper is increase, and then emia is in the blood. So hyperemia. So what would cause an increase of blood flow to the tissues? Well, your tissues are more active, right? So your blood vessels know that they need to dilate to give that tissue more oxygen. And it's signaled by that exact mechanism. So when you increase your metabolic rate, your level of oxygen is going to be used in the cells immediately. So you're going to have a low oxygen concentration, and then you're going to create a really high CO2 concentration, right? Oxygen's being used up. That's going to decrease the oxygen concentration. You're going to pump out a lot of CO2, the waste product of cellular metabolism. That's going to build up. And then the smooth muscle in your blood vessels are responding to that. So they can actually dilate, and that way they can supply the blood with more oxygen, and they can remove that excess carbon dioxide. So that is called active hyperemia, okay? So if you go through these pictures here, we can see that the dilation is going to promote increased blood flow, increased oxygen delivery, and then increased carbon dioxide removal. The difference between active hyperemia and reactive hyperemia is that reactive hyperemia is in response to the blood was shut off for a while. And so think about, for example, the blood pressure cuff example. you're pinching off a blood vessel, and the blood flow is restricted to some cells. So here are some cells. I'm just going to make a collection of cells. And then here's the blood vessel that's delivering blood to those cells. So if you stop the flow of blood because you've pushed down on that blood vessel, what's happening is that these cells are going to continue to be alive. They're going to continue to use oxygen, and they're also going to continue to pump out CO2. So what happens is that these cells are going to act in a similar way as exercise. They're going to use up all their O2. The O2 goes down. The carbon dioxide and metabolic wastes are built up. And because there's nothing to take the CO2 away, there's no blood flow to deliver more blood. So we have this instance where it mimics like you're exercising. So you have a lot of metabolism, and so you have and then when you release that blockage, then the blood vessels dilate because it's the same conditions as if you were exercising. They're going to dilate, and so they're going to quickly perfuse that tissue with blood, bringing oxygen in, removing the carbon dioxide. So that's reactive hyperemia. Okay, so let's just go through the flow chart real quick. So active hyperemia, you're exercising, let's say, you're increasing the metabolic rate, you're increasing oxygen consumption and CO2 production, and you're going to decrease the oxygen concentration, and you're going to increase the carbon dioxide concentration. So that's going to prompt the smooth muscle locally to dilate and to decrease the resistance, and that increases the blood flow, and that's going to deliver oxygen, and it's going to help remove the CO2 to bring everything back to normal concentrations. It's going to be exactly the same here, right? So everything is the same except for that blood flow. The initial cause is that we have stopped the blood flow. And then you stop the blockage, and then the blood flow comes back. And then the blood flow comes back to this this mechanism here, right? We have low oxygen and high CO2. So very, very similar, but the causes are different. All right, let's move on to the myogenic response. So myo means muscle. And so this is going to be a stretch of the muscle. So your smooth muscles can actually detect the stretch. And this is a characteristic inherent of smooth muscle. They actually have these stretch activated calcium channels, and they open and close in response to the stretch of the muscle. So if we follow this flow chart, right, we have high perfusion to the high perfusion pressure. That means that a lot of blood, perfusion means the blood coming to the tissue. So we have a lot of blood coming into this arterial, this small blood vessel. This large amount of blood stretches that smooth muscle on the wall. As a response, because of those stretch calcium-gated channels, they are going to constrict. So you're going to have muscle contraction. And that constriction is going to increase the resistance, right? Because all this blood's coming in, it's stretching too much, stretching those smooth muscle cells of the blood vessel, and that's going to cause a constriction. So it's going to then make it more resistant for blood to come in, makes it harder for blood to come in, so it's going to limit the amount of blood coming in. So the flow of blood coming in is going to be low. It's going to decrease. Okay, so it's going to negatively feed back, and then we can start to increase the flow again. But this is a response to the perfusion pressure. So there's a lot of blood coming in there. The muscle cells will actually contract to increase resistance. And the purpose of this, okay, in letter D in your outline, is to keep your blood flow constant and auto-regulate the blood flow through your blood vessels. There are other mechanisms here. So there are chemicals that can also act locally on these blood vessels. Oxygen, okay? So if there's a high amount of oxygen in the area, your blood vessels can actually constrict, which means that, it tells the blood vessels that there's enough oxygen present that we can actually get smaller. We don't need to be dilated to perfuse the tissue because there's already plenty of oxygen there. Carbon dioxide, if you build up in carbon dioxide, it's going to dilate those blood vessels because it tells the tissues that it's time to get rid of those waste, right? And it's time to probably deliver oxygen to the cells because the cells are very active. They're producing a lot of CO2. Potassium ions. So this is interesting. Why would potassium ions dilate your blood vessels? Because potassium is an ion that is going to be released, especially in muscle, with repeated depolarization, right? Because remember how your muscles or any cells have a higher concentration of calcium, potassium on the inside. When they are undergoing action potentials or depolarizing, they're going to lose calcium to the outside, to the environment, right? So potassium. So extra potassium ions in that extracellular fluid is actually going to trigger dilation because it tells the blood vessels, that these cells are very active and they're going to need more oxygen. Acids, are going to also dilate. And this is, if you think hydrogen ions, but also with carbonic acid. So as a metabolic byproduct, we know we can get lactic acid, and when you CO2 combines with water, you get carbonic acid. Okay, so a sign that cells have been working hard is this buildup of acid, either by CO2 or by lactic acid. And so that means that the blood vessels should dilate because it should deliver more oxygen to those cells because they're working hard. And they should also take away the waste products. And then lastly, nitric oxide. So nitric oxide is, we mentioned it before in the nervous system and in the cardiac chapter, that nitric oxide is a neurotransmitter, but it's also just this chemical that signals those endothelial cells. It says signals for vasodilation. It's a powerful vasodilator. It's also responsible for vasodilation and erection in the reproductive system. So it causes vasodilation, and that's just another factor there, and that's released by the endothelial cells themselves. And next one is just exercise. So during exercise, this is just showing you that there are independent regulation of blood flow. So the distribution of blood does not increase proportionally, right? So when we're exercising, our muscles do get more blood, but not all of our organs get more blood. As we talked about earlier, we don't have enough blood for all of our organs to get all the blood at the same time. So what's going to happen is you have sort of our resting distribution pie chart, and then we have our exercise distribution pie chart. So we can see that when we're exercising, our skeletal muscle gets the lion's share of the blood flow, and then all the other areas of our body, what's happening there is that the arterials are creating more resistance. They're constricting. And so that way, the blood that's coming from your aorta, the natural flow where there's the path of least resistance is going to be to the muscles. And that's why it goes to the muscles. All right. Extrinsic control. So outside of the blood vessels themselves, what can control the contraction and dilation of your arterioles is nervous system and hormones. Okay, so the sympathetic nerves is going to go to your smooth muscle, and the smooth muscle receptor here for norepinephrine is going to be called alpha adrenergic. So alpha receptors. And so if we keep it up here, so it's going to cause vasoconstriction. It's going to increase blood pressure because when you close down the lumen to constrict your blood vessel, that means that there's more pressure. It's a smaller tube, so the fluid that's in that tube is going to exert more pressure on those vessels. So it's going to increase blood pressure. Any vasoconstriction is going to increase blood pressure. So the sympathetic nerves will do that. And then we have three hormones. We have epinephrine, angiotensin II, and vasopressin. So epinephrine, it depends on the receptor type for what it does. So if it's an alpha adrenergic, as we have here, alpha adrenergic, it's going to constrict. So this is going to be on blood vessels. It's going to constrict the blood vessels in most of the body. But some areas have beta 2 adrenergic receptors, and it does the opposite job. And the places that would have an opposite response is your skeletal muscle and your bronchioles. Oh, cardiac muscle. And then also bronchioles. But I didn't write that down, but bronchioles too. In your lungs. Because if you think about it, this epinephrine is a sympathetic response, right? It's fight or flight. So it's going to raise your blood pressure. Raising your blood pressure is going to increase venous return. We saw in the cardiac chapter that venous return, if you have more blood coming into the heart, more blood gets pumped out of the heart. It gets pumped out more forcefully, and there's more of it. And so that's going to increase the circulation to all of your cells in the body, which is what you want in a fight or flight response. However, so for most of the part, your blood pressure is going to go up with vasoconstriction. But in certain areas of your body, skeletal muscle, cardiac muscle, and your bronchioles and lungs, you want actually dilation. You want more blood to go there. So this is why that they have a different kind of receptor to epinephrine. And when epinephrine binds there, it does the opposite job. It opens up those blood vessels, allows more blood to go there, because those cells, your skeletal muscle, your lungs, and your heart, are going to want to work very, very hard. So you need to dilate those vessels there. And also the bronchioles, the lung, the smooth muscle, there's actually smooth muscle on those airways of the lungs themselves, and you want more air to reach the lungs, and so those airways will open up and dilate. All right, so moving down to vasopressin. So vasopressin is a hormone. You guys also know it as a antidiuretic hormone. And it's going to also constrict the blood vessels, and that's going to increase blood pressure. So vasopressin is something that's going to increase water reabsorption. The more fluid you have, the more blood pressure you have, and then it's also going to constrict the blood vessel, which also increases the blood pressure. MAP is mean arterial pressure. Angiotensin II, also a powerful vasoconstrictor by itself and increasing blood pressure. So you should know this chart here. Capillaries and venules. Moving on to capillaries. So capillaries, we have a lot of capillaries in the body, 10 to 40 billion. Total surface area of 600 square meters. And most of our cells are within only 1 millimeter of a capillary, and most capillaries are just about 1 millimeter long, so we have many, many beds. Their walls, of course, are very thin. Molecules flow right through the walls, and they have the slowest velocity, so of blood flow blowing through them. They are very, very slow. So let's look at the anatomy of your capillaries first. So this is just the flow, the velocity. How fast does blood flow through your capillaries? So we're looking at the speed. So this is your velocity, the speed versus the cross-sectional area. Since your capillaries have the smallest diameter, the smallest cross-sectional area, we can see that the cross-sectional area is here. So then we have the velocity. We can see the velocity drops way, way down. So the smallest cross-sectional area is going to correspond with the distance, with the velocity, the slowest velocity. All right, so the capillary types you have, this is review from anatomy: continuous, fenestrated, and sinusoidal, or sinusoid. Continuous is the most common in the body, and we're going to have our endothelial cells connected together. We have a fully formed continuous basement membrane. We do have spaces for molecules to move through. Very small molecules. These are just metabolites, gases. Nothing big like proteins are going to go through a continuous capillary. Fenestrated capillaries are going to have regular fenestrations, and fenestrations are pores. And these are not as common. They're going to be found in specific areas of the body, such as the kidney and the pituitary gland, the surrounding the pituitary gland, so that it allows proteins to move through. So these fenestrated do allow proteins to move through. And then we're going to have our the most leakiest kind of capillary is called a sinusoid. And you probably remember that word from the liver. And this kind actually also allows cells to pass through, because you can see that we have really huge gaps within the cells. The basement membrane is incomplete, and so it's almost like barely there. So that's a sinusoid. So these are the 3 different kinds of capillaries. And for the most part, we'll be talking about continuous capillaries. So here is our microcirculation image. We have our arterials branching to a capillary bed, and then that capillary bed then leading to a venule. So let's look at our meta-arterioles. So this meta-arteriole, let's look at it, right? It is an intermediate between arterioles and capillaries, and it's going to directly connect your arteriole to a venule. So we can see that if we follow this, right? So if we follow the blood flow through here, here's our arteriole, it comes in, goes right to our venule. Now, these meta-arterioles can bypass capillaries. Because if you look at, let me see, I'm going to try and get rid of this mark. I think I have to do that by, all right. So if you look at the bypass capillaries, so it can act as a shunt to bypass your capillaries due to these smooth muscle rings in specific locations. So take a look at this ring here, this muscle here. So if this were contracted, the blood wouldn't go through that meta-arteriole. And so it would bypass the entire capillary bed. So it can contract and relax in response to local factors. When you contract it, the smooth muscle at the base of the meta-arteriole, it increases the blood flow through the capillaries, because it's going to stop and restrict the blood flow through the meta-arterioles. If it relaxes, more blood enters the meta-arterioles and it can go through the capillaries. I think this should be moved a little bit, anyways. So that's one factor that controls blood flow through your meta-arterioles. And then we have precapillary sphincters, which we can see here. And the precapillary sphincters are going to surround the capillaries. So they're going to contract and relax, and when they contract, they're going to stop blood flow through the capillaries. When they relax, they're going to allow blood through the capillaries. All right, so this is on a micro level, right? Again, this is microcirculation. We can control the blood flow through at these microscopic scale. All right, so exchange of materials across the wall of capillaries. How substances move across the capillary wall is through diffusion, and it's through transcytosis. Most of it is through diffusion. It's the most common mechanism. If a substance is lipophilic, so it is fat soluble, it can move right through the plasma membrane. So we can see in our picture here, gases, CO2, O2, fatty acids, steroid hormones, those are all lipid soluble and they can diffuse right through the membrane. Things that are not lipid soluble, that are lipophobic, right, they have a charge, they actually have to squeeze their way through pores in that capillary. And then the continuous capillaries are not going to allow proteins through. They're too large to go through the wall. So in order to move proteins through, you're going to have to undergo something called transcytosis. So transcytosis is, we looked at this before, but basically the capillary is going to create a vesicle on one face of the endothelial cell, transport it through a vesicle across the cell, and then export it through exocytosis on the opposite side. So that's transcytosis. All right, and then just the clinical note here, transcytosis has been linked to infections, because if this is not a very specific process, and if we were to transport, let's say, a toxin of bacteria, or maybe even the bacterium itself, the process of transcytosis can actually allow things like Chronobacteria Sakazekii into the intestinal epithelium. It can also move Listeria monocytogenes into the intestine. E. coli, the Shiga toxin created by E. coli, can also move into the intestinal lumen. So it can move from, it can just transport microbial products or the microbes themselves through the capillary membrane. So that is something that is part of microbiology, but this is due to transcytosis. Okay, moving on to bulk flow. Moving across the capillary. So we have pressure gradients as we move through the capillary. So what we want to look at here is that there's the arterial end of a capillary. So we're looking at capillaries only. There's the arterial end where the blood is still kind of moving according to a pressure gradient. Then in the middle of the capillary, we have no movement of molecules through, and the pressure is pretty much static. And then the venous end of the capillary, we actually get fluids to re-enter the capillary. So the arterial end of the capillary, what happens is we're looking at two kinds of pressure here. We're looking at hydrostatic pressure, which is the pressure of the blood itself. And we are looking at osmotic pressure. And it's called oncotic osmotic pressure because that's the term used for osmotic pressure created by proteins that are in the blood and proteins that are in the interstitial fluid. So because of these proteins, we have an osmotic pressure, and it just has a special name called oncotic pressure because we're using proteins. Anyways, it's just osmotic pressure. So at the arterial end, what happens is the fluid pressure is higher than the osmotic pressure, and so the fluid is going to exit the capillary since that hydrostatic pressure is going to be greater than the osmotic pressure. So there's a net filtration happening. So fluid leaves the capillary at the arterial end and contributes to the extracellular fluid. Okay, and in the middle there is no net movement because the fluid of the capillary, the hydrostatic fluid, is equal to the osmotic pressure. So they're both equal. There's no net movement. On the venous end, it switches where the hydrostatic pressure falls and the osmotic pressure pushes on the capillary. So you have a net reabsorption. So it's not absorption, it's reabsorption. So that's how that would work. So let's just take a look at a couple of examples here. Under normal circumstances, under normal blood pressure, we have the hydrostatic pressure is going to be highest at the arterial end and is going to fall in this manner as we reach the venous end, the venule. The oncotic pressure, the osmotic pressure, doesn't change because we're talking about proteins in the blood, we're talking about proteins in the fluid, and this is a very tiny space, so that pressure doesn't change. So that pressure stays constant throughout the little capillary here. So as we saw, when the hydrostatic pressure is greater than the osmotic pressure, we have a net movement out. We have a net movement filter out. In the middle of the capillary, they're kind of even, so there's net movement. And then in the venule end, we have our oncotic pressure or the osmotic pressure greater, right? This is greater than the hydrostatic pressure, so we have reabsorption. But what if we have high blood pressure? What if we have hypertension? 180/90. So if that happens, what is that your blood pressure is high all the way through the capillary, and the oncotic pressure doesn't change, or the osmotic pressure doesn't change. And so what happens is this scenario, right? So all along the capillary, you have the hydrostatic pressure is higher, so that you're going to get net movement out on both ends. That's going to lead to, you're going to add to that extracellular fluid that's going to build up, and that could lead to edema, which is swelling. If you have low blood pressure, hypotension, let's say that's, I don't know, just hypotension, 90/50, something like that. What happens is you have low hydrostatic pressure. Your oncotic pressure doesn't change. And all along the route of your capillary, we have the scenario where we have here that the oncotic pressure is greater than the hydrostatic pressure, and we're going to get net movement of fluid into the capillary, right? And that is kind of dangerous because your tissues are not going to get the oxygen that they need because or the nutrients and things that they need because we don't have that driving force out, we have a driving force in. All right, so hopefully that makes sense to you, some examples of those two pressures. And this is so this is our bigger picture of transcytosis, so it doesn't have to be so small, right? So transcytosis is endocytosis on one end, moving through the wall of the capillary through a vesicle, and then exocytosis on the other end. All right, moving into our next topic here, distribution of blood in various portions of the cardiovascular system. So we're kind of getting into veins, but before, I'm just going to check this. So before I talk about veins, let's just go through the rest of this topics here. So factors affecting filtration, absorption in the capillaries. So what factors can control outside of hydrostatic pressure and outside of the osmotic pressure control the flow of fluids in and out of the capillary? So gravity, standing on your feet, right? It increases the hydrostatic pressure because now you have gravity also pulling on the fluid, increasing that hydrostatic pressure. Injuries, when your capillaries are damaged and they're leaking fluids and proteins, that's going to cause swelling. Histamine is also going to increase capillary permeability. So histamine is released during injury and allergy. So histamine is a chemical that causes vasodilation. It causes the capillaries to get leakier and more permeable. Liver disease. So liver disease would actually cause a change in oncotic pressure because your liver is responsible for making albumin being the major protein that we have in our blood to maintain a proper osmotic gradient. So if your liver is damaged, you can't make albumin, and albumin would affect your osmotic pressure. And then the heart, if you have heart disease, you have pulmonary edema, swelling of the lung tissue, because your heart is not pumping the appropriate amount of hydrostatic pressure into your lungs. Okay, let's talk venules. Let's move on, I think, to veins. So in your veins, they are going to house the most of your systemic blood supply. So 60% of the blood in your body are found in your veins and venules. And you can see how this is in a pie chart out, right? The next highest amount is in your arteries and arterials, and then we have the pulmonary system, and then we have the heart itself, and then our capillaries. So the veins really hold a lot of blood. So how do we get all this blood back to the heart? This is a curve showing how the volume of blood in your arteries and veins varies with the pressure inside of them. So this is descending pressure. So if we take a look at our volume, so we have a volume of fluid, and we can see the pressure on the arteries, right? The pressure on the artery is here, and the pressure on the vein is here. So we have low pressure on the vein and higher pressure on the artery at the same volume. If we look at volume 2, we have the pressure on a vein and the pressure on the artery is the same. So how can we interpret this? It means that the veins are able to accommodate the higher volume without accommodating a lot more pressure. And so the veins are going to be able to accommodate more blood. And the way that we move our blood in our veins is with skeletal muscle contraction, which is called the skeletal muscle pump, and also with breathing, which is called the respiratory pump. and a couple other mechanisms. So this is review from anatomy, most probably, that every time you move your body, the thinner walled veins are going to collapse. And when they do that, they're going to push the blood inside the veins. And they're going to push the blood in only one direction because it cannot go backwards due to those valves. So the valves ensure a one-way path to the heart every time you move your muscles. So the respiratory pump, which I don't have a picture of, but it's the same concept with breathing. So when you breathe in, you inspire, it's going to decrease the pressure in your thoracic cavity, increase the pressure of your abdominal cavity, and the pressure on the veins in the abdominal cavity will create a gradient, and it's going to push the blood into the thoracic cavity, push the blood up, because you have less pressure because your ribs are raising, your thoracic cage is opening up, getting bigger, and that's less pressure. And then but at the same time, your diaphragm contracts down onto your abdominal pelvic cavity, creating more pressure. So that upward pressure, you have pressure in the abdomen, less pressure in the thoracic cage, so it's going to prompt that blood to move up. So it increases venous return by breathing in more specifically. All right, so the blood volume also. So what we looked at here was a respiratory pump. So we have the muscle pump, the respiratory pump, and then we're going to have the volume itself, and then we're going to have tone to the muscle themselves. So blood volume is first in your outline. So if you increase the blood volume, you're going to increase that venous pressure, and that's going to increase the blood moving through the veins. If you decrease the blood volume, you decrease the pressure. And this is a long-term way that you can regulate blood pressure by increasing the volume of your blood. Long-term, meaning that it's acting on hormones, something like vasopressin, to increase your blood volume. The last one is venomotor tone. This is the smooth muscle tension in your veins. So your sympathetic fibers are going to send messages to your smooth muscle of the veins, because there is smooth muscle there. It's just not very robust. And it's going to contract the smooth muscle, and that's going to be called tone, venomotor tone. And so if you, in response to the sympathetic nervous system, it's going to kick out norepinephrine. It's going to act on those alpha adrenergic receptors. It's going to cause constriction. It's going to increase that venous pressure, and it's going to decrease compliance. So it's not the veins are not going to be as elastic, right? If you have a more taut muscle in the wall, you're going to have less compliance, and that's going to be more pressure. It's going to increase venous return. So the idea is that when the sympathetic nervous system is firing, the walls of your veins become more taut, more constricted, and that increases the tone, increases venous return. So if you follow the flow chart, right, this is increasing the tone, decreasing compliance, increasing the pressure. So more blood increases the pressure in your veins. The tone increases pressure. The breathing rate and muscle pumping increases pressure. That's going to help your venous return, move the blood back to your heart. When you have more blood coming back to the heart, we looked at it increases your cardiac output by ways of increasing preload. And so that's going to overall increase your blood pressure, mean arterial pressure. All right. Lastly, with the lymphatic system is going to pick up the excess extracellular fluid, the filtrate. So although your venous end, the venule end of your capillaries does take back fluid, it doesn't all get brought back. So your lymphatic system picks up all the extra fluid. And the capillary network is next to your venous network. It's more porous. You should remember some of the anatomy from your class, but they're going to take that fluid, move it into the veins, at which point it's called lymph, right? So lymph is the fluid that's in your lymphatic vessels. It's going to run through at least one lymph node to screen your lymph for pathogens, and it's going to move that lymph towards your heart. So it's going to return to your bloodstream, to the venous blood, and that venous blood is going to, you don't need to know exactly where it comes into, but it's right before the heart, right before the right atrium. All right, so the lymphatic system has two jobs. The first job is to balance the fluid, this excess extracellular fluid, move it back to the bloodstream. And then the second part is the immune system, which we won't look at for a while. Okay, let's review. So which would not allow many proteins and large molecules to permeate other cells? So out of your choices, the capillary that does not allow proteins through is continuous. The largest pressure drop occurs across which vessels? So this one is arterials. Each day, approximately 20 liters of fluid is filtered and 17 liters is absorbed, meaning 3 liters of fluid goes from capillaries to the interstitium. This is extracellular fluid. What keeps the fluid from swelling the tissues? And this is lymphatic system. By moving skeletal muscles and by alternating pressure on the abdominal and thoracic cavity by breathing, you pump blood in the veins through their valves back towards the heart, which is not a result of increased activity of these pumps. So if you look at your choices, it will increase venous pressure. It will increase the mean arterial pressure. It will increase the end diastolic pressure. So the only thing that does not, the pumps of your skeletal muscle and your breathing, it does not increase tone. That is your sympathetic nervous system.